Cleveland Clinic there. He's going to be talking a little bit on tissue reengineering using this extracellular matrix. And it's this was presented at our meeting last January at the SIS. It's a very intriguing process in terms of what the body and the cells are capable of doing, and especially as much as for what it can do now, but what I think the future is going to be bringing to us in terms of the potential of the cells in our body to regenerate tissue. It's also being used, by the way, cardiologists are using this for now for closure devices. We're not going to be discussing that.
The topic about today is myocardial regeneration. And essentially, to simplify, what we are made of are two things, and that's cells and extracellular matrix. And this has been described actually a quarter of a century ago by Mina Bissell, who was at Berkeley. And what she described essentially was how does matrix express gene expression. I think it shows the intimacy of the relationship between the cells and the matrix. But essentially what she talked about was a dynamic reciprocity between the matrix on one hand and the cytoskeleton. So that the matrix is postulated to exert physical and chemical influences on the geometry and biochemistry of the cell via receptors. This woman is up for a Nobel Prize now. So this is a quarter century after she first described this. So she was on to something. We talk about tissue engineering. Normally the constructs is very, very complex. People have a bit of a matrix, they try to seed cells on something, they take tissues and they put them in a culture or bioreactor. This process is very time consuming and very, very expensive, about $20,000 for a single vascular graft. So the idea is, what if you could take the matrix, attract the body's own cells, put the matrix into the body, and allow the body to act as the own bioreactor. And that is the point of what we're trying to do with matrix, to short circuit that very, very complex. And I guarantee the more that we are involved in this part, the more we'll be able to screw up nature. So hopefully the body will do a little bit better than we can. The components of tissue engineering are really three main things. It involves cells, the matrix itself, and signals. And we're all very, in cardiology, we know very, very well how trying to regenerate heart muscle and so forth. VEGF was big for a while, stromal-derived factors, trying to get signals, put signals into infarcts, try to get cells to go where you want them to go. So there's been a lot of attempts to try to blend these three areas in engineering. So it's fairly daunting. If you're talking about matrix, what is it going to have to do to regenerate myocardium? So what are the processes needed to regenerate myocardium? So let's go through. First of all, when the stem cells go to the defective site, they have to know what to go into. So if a stem cell goes in and turns into brain tissue, that's not going to be very good. One of the issues is the cells have to connect and speak to each other. There's every crosstalk. Myoblasts go in and they form little islands. They haven't been communicating with other cells or whatever. So they have to establish cell-cell connections. One of the big things is you have to establish a vascular supply at the site. So if you put a stem cell in an infarcted zone, where is it going to get its nutrients and its angiogenesis? So that's going to be a problem. The other thing is when it goes in there, it's got to turn into the tissues that it's intended to replace. The other thing is, and one of the concerns with embryonal stem cells is you got to why don't you create tumors and stuff? You got to limit them. Once the tissue is made, it's got to know to stop. Then, in cardiac specifically, you got to couple electronic conduction. So, in other words, one of the problems is when you have myoblasts, when you injected them in, they have their own little electrical milieu causing arrhythmias. So the electronic conduction has to be coupled, and you have to establish matrix connections by way of cell adhesion. So the tissue that you're replacing has to connect with the other tissue that's already there. So it's a pretty daunting task for what you're asking to do for myocardial regeneration. So what is the role of the matrix scaffold that we're going to try to use to do this? Well, what we're hoping it'll do is organize cells into tissues by endogenous recruitment. Okay, so that means that we put it in and we expect the body to take care of populating that scaffold. The next thing it's got to do is once the cells get in there, it's it's got to coordinate the function. It's got to tell them what to turn into. It's got to get them to communicate. It's got to get them to act together as a complete tissue. The next thing is it's got to supply the infrastructure. It's got to allow and provide the normal metabolism. Heart cells are very, very metabolically needy. They need a lot of garbage, fuel in, garbage out, and so forth. The other thing is there's got to be crosstalk between the cells that go in and the native tissue. But more importantly, we're asking them to actually differentiate properly, proliferate and grow, and even express the same gene. So what we're asking the matrix to do is what Mina Bissell said. We're asking the matrix to drive the phenotype of the cells that are injected. And this is what it looks like at the end. The human extracellular matrix is a three-dimensional structure. And that's one of the things when you're plating cells out on flat, you don't get that three-dimensional architecture. That's one of the things we know that's very important. When stem cells come and populate a matrix, we know that it's sort of a lock and key phenomenon. The stem cell will mold and differentiate and then phenotypically express itself variably depending on this lock and key mechanism. Here, there's muscular lacunae and capillary lacunae essentially. But one of the things we really know is that the role of the extracellular matrix is very, very important. When the matrix degrades, cells die, okay? No matrix, cells can't survive. And all the cytokines and so forth that go along with that. Circulation 2008. I'm just going to emphasize this. One thing we know from matrix biology is that a cell cannot survive if it's not attached to a healthy local matrix. If the donor does not have a healthy matrix, it simply won't survive. So, based on this, I'm going to tell you, you can inject through a catheter any kind of stem cells you want into an infarct. It isn't going to work, okay? If you put stem cells alone, or VEGF alone, or whatever it is, it's not going to work. It's not going to work based on what we know now about the very important relationship between matrix and stem cells. So, we know that when we do inject these cells, and the cardiomyocytes are already damaged and so forth, it will not engraft. And that's why when you see the studies that have been done on injecting of stem cells to date, there has been no demonstration of true myocardial regeneration. The stem cells that do stay in the infarct area populate the rim of the infarct only, where the matrix is healthy, where they can get fuel, and then, but why is there a positive effect? Well, the positive effect, guess what, is a paracrine effect. And that, the effect of the stem cells that do go, they exert a paracrine effect, which essentially, they secrete substances that down-regulate something called MMP, matrix metalloproteinase. So guess what? The stem cells that do survive when we inject them are actually having an effect on the matrix. So, what is in this magical matrix? Well, it's made of a lot of things. Primarily, it's collagen. Type 1 especially. There's also a lot of other things. Glycosamine, glycan, glycoproteins, fibronectin, all these big worded named things, okay? I had to go back and sort of read what they were. I forgot what they all were because I didn't really pay much attention. But there's a lot of stuff in there and growth factors. The bottom line is, okay, if you're trying to regrow heart muscle, unless you have all these things, you're going to fail. Okay, so you would say, okay, let's try VEGF. You stick that in. It's not going to work. Let's try VEGF and homing factor. That's not going to work. Let's try, the problem is, it's all these things together that impact cell differentiation.